Blue sheets, blue sheets, blue sheets. Now, if you hear that, you're a student here at my school, whether in my class or my colleague's class, we let them do these handouts where they fill it out. And so I want to do a little video on showing some student work with the blue sheets, and we'll first talk about how we do it. And as usual, brought to you by Curious Maryland and the Rubiscokid. So, years ago, my friend Jason Schweitzer and I started teaching, we AP biology together, and then he was like, "Hey, you want to do anatomy?" So in 2008, he set up the course, and so what I want to do now this year is kind of some videos and show how we've changed our approach and how we've gotten students to maximize learning and to learn the material, not memorize. So first, as I said, brought to you by Curious Maryland, I want to show you what exactly is the directions that the students get when it's time to do blue sheets. If you're familiar with physics where they made you do a handout that you can use, it's a note sheet, it's a reference sheet. I hate to use cheat sheet, but it's a reference sheet. And I should point out, over time, we've usually require them to do blue sheet, but this year we've changed color because of running out of paper and stuff. So this is the general handout that the students follow. The day of a test, they must turn it in 10 minutes before school starts. And if they don't do it, they don't get to use it. They want to focus on concepts. And I want to switch to another handout to show you the rules for this particular test that they took on epithelial. So we are pretty transparent. We ask what we say we're going to ask. So this was more of a review guide or a study guide, if you want to say, and you're going to have to be able to describe, you're going to have to identify, you're going to have to connect. Well, that's a huge one. So I'm going to do another video on specific stuff that we did to help the students be better prepared, and they did pretty well. So the purpose really for this video is to show student work and show you some of the blue sheets. And by the way, I know I can sense the teen species. I've been dealing with them for a long time. They're probably rolling their eyes if they're watching this, hearing me sing the song, because I yell it out and have them try and sing it to me. So, all right. So let's do a couple different variations. So first, we let them put anything they want, but it's not anything they want, it's anything they need to show that they know it. And the reality is, it's the classic case. Most of the students who do this level of work do really well, and they don't even use it. And that's kind of what the big trick for years started. I remember with physics teachers, they're like, you put so much time into it that you don't even know it because you're not memorizing it, you're learning it. There is a difference. And that's a big thing that we've been trying to stress, is connecting the learning. So this is a good one. This student showed the mucosal layers. They showed pretty cool stuff. They showed an endocrine and exocrine variation, starting with a simple cuboid cell and doing this. This was an exercise that we actually did in class, and I meant to show it, but oh well. Um, so let's do a couple others. I'm going to zoom in and zoom out. And wow, you get some brilliant work with these students here. So it doesn't have to be this year. It doesn't have to be on blue sheet paper, even though it says it in the directions. We realize that we're running into an issue. I mean, when you give students a chance to go above and beyond, that's one of the rewards of teaching, is getting them to go beyond what they thought they could do. All right, so this kind of shows, I like this because it stresses a concept that the Rubiscokid, Jason Switcher, myself, started years ago. And he started, hey, they got to be able to visualize. And he's very good on that. And I mean, you're going to see a lot of videos this year of us showing those visualization exercises and those connections to learning. So over here, you see written description. Over here, you see the cartoon picture. And one of the things we stressed was you should be able to see a picture and write a written description of it. Simple declarative sentences is in some cases. All right. Or you should see picture information here and draw a picture that represents that. I mean, this is like amazing work. All right. And so in this particular case, and let's try and connect it to one of the things we expected them to do was be able to draw some epithelial cells and show everything in there. I mean, you see that little I mean, this is like textbook level animation. So, like I said, like I love when we get students to go above and beyond, showing Desmond zones, embedded proteins, adhesions, these cells connecting together. Because epithelia has very tight spaces. They're tightly packed. There's a little space in between what we call matrix, like what we'll see with connective tissue, which has more space in between cells. So I actually did allow some students to pick these, and this student made errors and got worried, so they did a separate sheet of paper and just taped it over, which is one of the things we allowed for them to do. Showing pseudociliated or pseudostratified ciliate columnar. I should know. I was pronouncing these things. One of the things they put here was the different types of epithelial membranes: serous, mucus, and mixed. They left off cutaneous, but all right. All right, so let's do a couple more. So this one's on purple paper. Let's zoom out for a second. Look at what we have here. So at the top, they described the 4 types of tissue. So epithelia, connective, muscle, nervous. So this particular test was just this with the introduction to histology. Then you got the perfect breakdown of epithelia tissue. You have the apical surface, you have the basement membrane, and then you have like cytoplasm, cytoskeletons, like protein. And a wise student would go through this. We're transparent. We ask what we say we're going to ask, and we prepare the students, and then they got to do their time. They got to do their time, and then hopefully it works out. And I will say, having graded this test, a lot of students did pretty well. All right, 5 characteristics, functions. Some students got bogged down between a feature and a characteristic. So that was one of the questions I was asked a lot about. They talked about solute solvents, basic parts of the cell membrane. This is from an exercise we did in class, so they already have this in their notes, so they were either transfer it. Right? So we put endocrine and exocrine. We started just with the simple cuboidal, and we formed this, and we showed endocrine is going to the bloodstream versus exocrine is going to ducts. I like that they put here the difference between stratified squamous and transitional. See, transitional is in the urinary bladder, and when you have a relaxed bladder, no urine, it's more circular shape. And then you have the flatter one because it's kind of like here's the circular shape, but when you have urine, it pushes it so it makes kind of a flat. So a flat cell versus the technical thing that we call, whoops, let me get this, a dome cell. And they have some stuff on the back. Now this one, they just did a chart. And again, we gave them permission to, okay, let's try and get in there. Now for this was a chart that they filled out where they had to show various organs, and then they had to say what type of tissue. Was it a lining or a covering? And we end up having them do this on the test, all right? I'm gonna have to take a picture of that and show it for the other video. Okay, more. These are some pretty good ones. There's this again. And what do they show here? They describe the 3 membranes. I mentioned earlier, the other one didn't mention cutaneous. So this was mucous, cutaneous, and serous membrane, showing the serous is open to the outside versus the mucus is lining. And endocrine, exocrine again. I'm glad to see a lot of them did that. Basic cell membrane, talking about cell membrane being permeable. Molecules are permeable or they're impermeable. But the cell membrane is what we call that selectively permeable. But it also has other aspects of it about cell communication, a bunch of other proteins, stuff like that. So they list the characteristics. They listed these, but they also define these. Again, being able to identify it and then look at the structure and tell its function, which is a key part of biology in general. This one again shows the difference between, so My focus is acting a little weird here. So one of the things, and this is literally from the notes that I gave them, so I'm happy to see them do this. One more time. All right, so their paper was pink. It looks like it's blue, oddly enough. So there's the stratified squamous. Here's the stretched transitional, and here's the relaxed. And one of the things we tried is look for the apical surface, and then when you're dealing with the difference between stratified squamous and transitional, they're flat all the way for a couple, and then they get circular towards the bottom near the basement membrane. But the stretched is flat here, but the very next cell is going to be circular, and then relaxed is a circular, and we call that a dome cell. All right. Couple more. So, like I said, I'm not going to spend so much time elaborating on every one. I'll just show you some of the good pictures. All right. Now, this is someone who is, all right, anyone who puts this amount of time probably didn't even look at this because they learned it, they didn't memorize it. You might know I keep saying that. That's one of the things that we want students to be able to focus on. All right, and they could make it any particular way they want. Depends on the test. We give them 2 sides or 4 sides. So they start by talking about the shapes and layers. The 2 discriminating factors for epithelia is the shape and if there's any type of layering. Then they wrote out descriptions and location. Description, location, function. Description, location, function. This is going to be very important for them to understand that we get to connective tissue, which is going to be more extensive, and of course the other 2 tissues. The main component of the apical and a basement membrane, or basement surface. This is the second one where they kind of did more pictures on one side and charts on the other. All right. All right. So again, look at this one. I got to zoom into that. Structure of actual permeability. They're showing the lipid bilayer. That's really good with some proteins in there. Characteristics. All right. And yeah, that's kind of wrapping it up. So they go from here to one of these sheets to success on the test. That's always what we want them to be able to do, to demonstrate what they know through a variety of ways. All right. Thank you very much. Brought to you by Curious Marineland.